Mitosis and Meiosis by Malcolm Owusu and Vishaka Joshi. Living creatures on Earth, like you and me, are constantly growing and reproducing. But how exactly are living things able to carry out these functions? Well, to explain that, we must start with the cell theory. Established by Rudolf Virchow and Theodor Schwann in the 1830s, the cell theory is comprised of three main principles. 1. The cell is the basic unit of life. 2. All living things are made up of cells. And 3. All cells arise from pre-existing cells. Eukaryotes are able to fulfill these principles through two processes of cell division, known as mitosis and meiosis. Mitosis is a form of asexual cell division that is seen in the production of somatic or body cells. It creates two daughter cells with chromosomes identical to the parent cell, also known as clones. Mitosis is important for growth, regeneration, and repair. Meiosis, on the other hand, is a form of cell division with the purpose of creating gametes, the egg and sperm cells, that are required for sexual reproduction. It results in the formation of 4 daughter cells with half the number of chromosomes as the parent cells. Now, let's look at meiosis. All cells go through the cell cycle, the series of events from the time a cell divides to the time the daughter cell divides again. In somatic cells, the cell cycle consists of 2 main stages, the first of which is interphase. Interphase has 3 stages, G1, S, and G2. During G1, the cell grows and differentiates itself. It builds up energy that will later be used in S phase. S phase is when DNA synthesis occurs. Chromosomes are thread-like structures made up of DNA that carry a cell's genetic information. The center of a chromosome is called the centromere. When a cell arrives in S phase, there is a single chromatid attached to the centromere. After it undergoes synthesis, an identical copy of the chromatid is created. This is known as a sister chromatid. At the end of S phase, all of the cell's DNA has been replicated. In G2, the cell prepares for division. It does this by making proteins, storing food, and duplicating the cellular organelles. Now the cell is ready for division. Meiosis is the process of creating 4 unique gamete cells. Meiosis consists of 2 rounds of cell divisions, one of which separates identical or homologous chromosomes, pairs, and another that separates a sister chromatid. The first stage of meiosis 1 is prophase 1. Here, the chromosomes in the nucleus condense and homologous chromosomes pair together. A process called synapsis occurs, where homologous chromosomes fuse together to form structures called tetrads. The fused chromosomes swap portions of the chromatids at sites called chiasmata to create new, distinct sets of DNA. While this occurs, the nuclear membrane disappears. Additionally, the centrosomes duplicate, migrate to opposite poles, and form the spindle apparatus, which connects the centromeres of the chromosomes. In metaphase 1, the chromosome pairs line up at the metaphase plate at the equator of the cell, connected to the kinetochore microtubules, similar to metaphase in mitosis. They line up independently of each other. In anaphase 1, the cell elongates as the non-kinetochore microtubules elongate, and the kinetochore microtubules shorten. Homologous pairs of chromosomes are separated and pulled towards opposite poles. In telophase 1, new nuclear membranes form around each cluster of chromosomes as they uncoil inside of the nuclear membranes. Animal cells lay down a constricting ring made of actin between the 2 nuclei. This contracts and creates a dent in the cytoplasm called the cleavage furrow. In cytokinesis, the actin ring pinches off the cytoplasm between the 2 nuclei, creating 2 new cells. Plant cells have a rigid cell wall and therefore cannot pinch off cytoplasm like animal cells. Instead, plants lay down a cell plate made by fusing Golgi vesicles between the nuclei, which separates it into 2 daughter cells during cytokinesis. Meiosis 2 comes right after meiosis 1. The first stage of meiosis 2 is prophase 2. In this stage, the spindle apparatus once again begins to form as the centrosomes move to opposite poles of the cell. The nuclear membrane disintegrates and the chromosomes condense. Microtubules attach to the kinetochores on the centromeres. In metaphase 2, the chromosomes line up in single file on the metaphase plate. Again, the order which they line up is entirely random. In anaphase 1, the cell once again elongates as the kinetochore microtubules split the centromeres and pull the sister chromatids apart. In telophase 2, New nuclear membranes form and the chromatids uncoil inside of the nuclear membranes. The respective method of cytokinesis occurs for animal and plant cells once again, and the cell is now divided into 4 haploid daughter cells. From here, there are 2 cases. In females, oogenesis will occur. Because of uneven cytoplasm division, 3 of the 4 daughter cells will be non-functional, and only 1 ends up becoming an X cell. The other 3 are polar bodies. Spermatogenesis occurs in males. All 4 daughter cells will have equally divided cytoplasm, and therefore all will become sperm cells.